We were talking about the teeth last time, and let's go ahead and finish up that topic. It's just now the anatomical structure that we're interested in. The part of the tooth that you see is enamel, and that covers the crown of the tooth. Or that area that you see, and enamel indeed is the toughest substance in our body, even stronger than bone. As far as the root, that is the area under, or part of the tooth under the gingiva. And when that gets inflamed, that causes gingivitis and puts you at risk for actually losing the tooth itself. Dentin, I want to go to that next instead of the cementum. Dentin, that is the bulk of the tooth. And so when you take a look at our picture here, all of that tannish or pale tan material is dentin. And so we have it in an area underneath the crown, and then we have it also in the root area. The important thing about dentin is that's what is the active part of the tooth. Inside is the more living pulp area, but cementum is that which covers the dentin that's in the root area. So the dentin is covered by enamel at the surface, and then the dentin is covered by cementum in the area of the root. And that is indeed calcified connective tissue, and is kind of the covering or the link between the dentin and the periodontal ligament, which is associated with the bone itself. Pulp cavity contains a substance that we call pulp, which is basically a loose connective tissue, blood vessels, and nerves. And so we know that a tooth is alive because you can have dental pain, and that is the pain of the nerves. And also, if you get a tooth knocked out, one of the things that you want to do is you want to grab that tooth and go to the dentist as quickly as you can so they can put it back in, because at the base of the roots, we are directly connected to blood vessels and nerves. And so if we can get that tooth in, get it properly situated where it's close enough to where it got sheared off, we can actually have regrowth of those blood vessels and even the nerves itself. Now, if that works, you can tell, because you can tell actually more about it if it doesn't work. Because do you guys know what color the tooth turns if they try to put it back in to reestablish connection and it doesn't work? Gray and black. So that actually means that there is no vascularity actually associated with the tooth, and the tooth has died and has undergone necrosis internally. And so even though you still see a crown externally, it's not enough to actually block the color transition inside. So that's an important part of our teeth. They are indeed living. What kind of joint is this? Is this a syn, anthi, or diarthrosis? My goodness, all over the place. So synarthrosis is not movable, amphiarthrosis is slightly movable, and diarthrosis is freely movable. This is a synarthrosis. And then it has a very special name too. It's a gomphosis, peg and socket. Yes, joint. Moving down the digestive tract, we come to the next structure, the pharynx, and that is indeed skeletal muscle that helps us with the swallowing process. That is also known as deglutition. That's in our notes. We'll get to that in a moment. Helps to propel the food downwards, and of course our soft palates covering up our nasal pharynx so that we can make sure it indeed just goes downwards. The esophagus is often referred to as the food tube. Runs down the mediastinum through the diaphragm, through an opening in the diaphragm called the esophageal hiatus, and then connects with the stomach down below. Now, when this is not being used, it is collapsed or flat. And that's one of the things that you guys noted when you actually were doing your laryngeal dissection, and you separated the esophagus at the area of the attachment of the laryngopharynx. And you noticed that it was kind of difficult to identify it because you probably thought the esophagus was going to be more tubular in its appearance. And it is a muscle, and it is a tube when something's inside it. But when nothing's inside it, there's no reason to have it open, because if it was open, you get air in the stomach. And so it's best to actually have it collapse to keep air from getting into the stomach for you. At the area where the esophagus joins the stomach, we call that the gastroesophageal sphincter. Some people will also call it the lower esophageal sphincter. Always waiting for that. And I prefer to call it the gastroesophageal sphincter. Our notes say it acts as a valve. Remember this, when we say valves are meant to help things go only one direction to prevent reflux, but it's very weak. It's our weakest sphincter. And we know this because in our society we have a pretty significant problem due to that weak sphincter. Acid reflux, heartburn, those types of things. And so the other thing to realize too, certain foods that you eat actually cause the smooth muscle in that sphincter area to actually relax even more and cause food to actually go up. Unfortunately, things like chocolate will actually weaken the sphincter for you. As far as the muscular arrangement, the upper 1/3 is skeletal muscle. The middle 1/3 is skeletal and smooth, not surprising. And then the lower 1/3 is smooth muscle. So it's an interesting arrangement of skeletal muscle and smooth muscle. Start skeletal at the top with very similar to what we have for our pharynx, and then start to transition into smooth muscle where we actually have just smooth muscle down there at the lower esophageal sphincter. The other thing too about the arrangement of the fibers, we have outer longitudinal and inner circular. And we already talked about the lining of that. The mucosa would still be stratified squamous epithelium because we still swallow, fish bones by accident, and other things are still kind of rough form sometimes when we're noshing quickly and swallowing quickly because we're wolfing our food down. It doesn't always go down in a nice smooth bolus. The esophageal hiatus is supposed to be only large enough for the esophagus. What happens if it becomes dilated or too big? You get a hiatal hernia. And what is actually herniating through that esophageal hiatus? Is the esophagus going into the abdominal pelvic area? Or is it something else coming up into the thorax? So it's coming up into the thorax, and that would be the thing that it's attached to, and that's the stomach. And so sometimes that also is a cause of significant heartburn for some people. And as you can imagine, if you had surgery on it, you could actually help with that process, making that hole smaller again so that the stomach doesn't protrude up through that. As far as our digestive processes so far, mouth, pharynx, and esophagus, there's no such thing as a digestive process in the pharynx or the esophagus. That's just a transit tube. In the mouth, however, we had mastication, which is chewing, and that is done to mix saliva with the food to form the bolus. And a bolus is a large volume of material that's moving into you. And with mastication, it does say chewing here, but you really don't need your teeth for chewing, because people who are edentulous without any teeth, they actually often prefer to take those darn dentures out when they actually have their meal, because they keep just falling down and moving around in their mouth. They're not stuck in place properly. And so they'll use their tongue on their hard palate in order to actually mash their food around to get it broken up into smaller physical pieces. Deglutition is the fancy word for swallowing, and then peristalsis occurs in the esophagus. Again, swallowing is movement of the bolus, and peristalsis is movement of the bolus down the esophagus. And so this peristaltic wave is actually going to be due to constriction of the longitudinal plus the circular fibers behind the bolus and relaxation in front. So we're able to squeeze that down that tube just like a tube of toothpaste. We want to actually move that in the proper direction towards the stomach, and that way you can do that whenever you're standing on your head or if you're standing upright. You will actually allow the material that the esophagus is moving down to the stomach, Stomach is designed to break the bolus down into chyme, and I liken chyme to the stuff that you see babies spit up. So when you have a breastfed baby and it spits up, it looks like slimy cottage cheese that's been mashed all up. And that's what we want to do. We want to make it into this slimy, mucoidy mess. And that's chyme. And it's going to be very acidic. If we could get the door closed back there, that would be great. Thank you. And so we'll talk about how we make that bolus into acidic chyme in just a moment after we go through the anatomy of the stomach. Anatomically, when you look inside the stomach, you will see rugae if the stomach is empty. If the stomach is full, you will not see rugae because rugae are internal folds that are designed to help with expansion of the stomach. After the esophagus comes in, there's an area inside the stomach called the cardiac region. It's a very small area. It's basically right adjacent to the gastroesophageal sphincter, and sometimes people will call it the cardiac sphincter. It is indeed quite small and involved in a transition area for you. After the cardiac sphincter, the next area that's usually discussed is the upward bulge, fundus, because we'll talk about fundus with the uterus, for example. The uterus has an upward bulge, and the stomach also has one, a large upward bulge. And then as we move on down the outer aspect of the stomach, that's called the greater curvature, we'll come to the bulk of the stomach, which is indeed going to be our body of the stomach. And then as it makes a turn and becomes narrower, that's going to be our pyloric region. So the pylorus is where the stomach makes a little turn and becomes narrower. At the end of the pylorus is the pyloric sphincter, and then after that, we're into the small intestine, the duodenum. Don't forget, all of these features of the stomach are meant to be, you should be pointing out when you do your digestive eval on the cat because they have all of these areas also. They have the greater curvature, and there's something attached to the greater curvature. Greater omentum. They have a lesser curvature, and there's something attached to the lesser curvature. Lesser omentum. Will you have destroyed most of the lesser omentum when you do your biliary dissection? Yeah, so a lot of that lesser omentum will be destroyed, but if you have a little bit, don't forget to point out when you point out lesser curvature that the lesser omentum was actually attached in that area. So those are the basic anatomical features of the stomach. Also, we have a nice histology view here, and I can tell, and as you guys realize you're going to have a histology eval in a little bit, don't forget to use some of these pictures as we're going along to help remind you the histology of an area. I can tell that this is the gastroesophageal junction. I can tell you that we're in the cardiac region of the stomach because I know that this is stratified squamous epithelium, right? And then suddenly it stops and we get into this area where we have these protrusions. Those protrusions are villi. So we have these finger-like protrusions, villi, that are covered with all kinds of goblet cells. And that's a very important thing that we're going to talk about, stomach physiology, because the stomach has to protect itself from what it does, which is create a bunch of gastric juice, which has high acid content, and pH can be down as low as 2 inside the stomach. And so you have to protect it, and that's what those goblet cells are all about. So do take a moment to look at the pictures as we go along histology-wise, and see that your histology in your lab composition notebook is also very similar. Let's take a look at the modifications that were made to the tunics of the stomach. And remember, tunics are layers or coats to the stomach. And on the muscularis externa, we still have our circular and longitudinal layer, which is the two that we've talked about previously. But the stomach is unique in that it has a third layer. So there's 3 muscularis externa layers, as we have an oblique layer. And this is designed to help the stomach churn. So the stomach not only does segmentation and peristalsis, the stomach acts like churn to help make that acidic chyme. Also, the mucosa possesses a columnar epithelium that consists entirely of goblets. And these goblets So you take a look, and the entire lining is goblet cells. Now, goblet cells are always associated with what type of epithelium? Simple columnar and pseudostratified columnar, correct? So, but remember, pseudostratified columnar is still simple columnar, correct? And so these goblet cells are still hanging out with the same cell types that we've associated them with in the past. But we don't see any simple columnar in this area because we need to protect this area. And what these cells do is they produce an alkaline mucus. We're making a very acidic gastric juice that's going to go into the lumen where the food stuff is actually located. And so we want to protect us with a mucoid layer that is alkaline, so that minimizes the ability of the acid to get down to the cells to destroy the cells at the cellular level. If you have a defect in their ability to produce alkaline mucus, what can result? Break down stomach wall and get an ulcer. And so when people have peptic ulcers, they're actually talking about there's been some mucosal dysfunction to where the acid can now start to eat away and erode. And that's not what we want. We want nice, healthy mucosa. The other thing about this, which is really kind of interesting, we have these things also that are called gastric pits. And it says millions of gastric pits leading into deeper structures called gastric glands. And you have to realize that you can only see these with a dissecting microscope. So you open up the stomach and you take a look at it, and then you get a dissecting microscope, and it looks like it's got little divots all through it. So millions of gastric pits are those little divots. They are also all lined with mucus cells. So there'll be mucus cells on the surface, mucus cells in the gastric pits, and all the way down into the gastric glands until we actually come to the cells that are producing gastric juice, which are our parietal cells and our chief cells. The parietal cells are the cells that produce hydrochloric acid and intrinsic factor. Intrinsic factor is vital for B12 absorption, which occurs in the small intestine and more specifically in the ileum. So B12 from our foodstuffs has to interact with an intrinsic factor, follow it, go on down the small intestine all the way to the ileum for actual absorption for us. And do you guys remember what B12 is vital for? It's vital for healthy red blood cell production. So parietal cells, they are big and they're fat, and they are more lightly staining. And then as you look on down the gastric gland, you'll find the chief cells, also called zymogenic. That's a very common name for them, zymogenic, so do keep that in mind. They produce the enzyme called pepsin. Now, it's produced in an inactive form, and once it gets into the lumen of the stomach where the acid pH is found, that activates it. Pepsin is a protein digesting enzyme. So requires low pH for activation. And we are going to see this as we go through the digestive system, that an enzyme will be produced in an inactive form, and then a pH change will cause its activation. And this is a safe way to make these enzymes so that you don't auto-digest yourself. So that's one of the things that's important there. There is also, and you can't see these guys without a special stain, these guys called enteroendocrine G cells. And they're called G cells, which is nice because they produce the hormone called gastrin, which is designed to increase gastric juice production. So gastrin increases gastric juice production. The more gastrin we have, the more gastric juice that will be produced. And when I say gastric juice or juices, I'm talking about hydrochloric acid and pepsin. So those are the main components of our gastric juice. So let's go ahead and move on down in our notes and look at the digestive processes involved in the stomach and realize that we can bring our pH in the lumen of our stomach all the way down to 2. That's a really very acidic pH. That will indeed start protein breakdown. And again, pepsin is most active at this low pH. The 3 phases of gastric secretion, in other words, gastric juice production, are the cephalic phase, the gastric phase, and the intestinal phase. So head, stomach, and then also gut or small intestine. It's important to realize the stomach will secrete 2 to 3 liters per day of gastric juice. So we already made 1 to 1.5 liters a day of saliva. Now we're making 2 to 3 liters a day of gastric juice. So there's a lot of fluid that's going into the lumen of our digestive tract. The cephalic phase. Cephalic is your head, and that's a cerebral phase because what it means is that anticipation is important in actually the release of gastric juice. So what are these things that will bring about the anticipation of food into your stomach? Well, that's the sound of bacon. That's the sight of bacon. That's the smell of bacon. That's just the discussion of bacon. I already gave you a cephalic phase just right now. You already have some gastric juice being produced by just the mere discussion of food. Haven't you found that, man, I really want to eat now. Just the thought of food makes me hungry. And indeed, you're pouring all those gastric juices in there in preparation for the arrival of food. Now, the gastric phase means that the food got there, right? And that's going to cause 2/3 of the total release of our gastric juices. And what's important for this is that hormone, gastrin, that is going to cause hydrochloric acid release from our parietal cells. And what activates this hormone release is stomach stretch. So that's baroreceptor related. So a neural mechanism will cause a hormonal release, which will cause a release of more hydrochloric acid and pepsin, which is kind of interesting. So that's that pressure receptor release. Now there's also a chemoreceptor response to stomach juice release, and that is the dilution of our stomach juices. So you have a whole bunch of gastric juice in your stomach and you drink a glass of water. What does that do to the pH of that gastric juice in the stomach when you add a bunch of water to it? Does it make it go down or does it make it go up? It makes it go up. And so we actually have chemoreceptors that detect those pH changes whenever you pour stuff in, whether it's fluid or food. Because when you pour food in, one of the things like proteins actually have a little buffering component to themselves, which will also cause the pH to go up. So anything that causes the pH to go up or become less acidic will also stimulate gastrin release. So dilution of stomach juices means that pH is increasing. It's still never getting to be very alkaline, but all of those things will cause gastrin release. So another thing that's interesting, stimulant for gastrin release, is caffeine. So there's even certain foodstuffs that we actually ingest that will also stimulate gastrin release. So that's the mechanical aspect that is causing gastrin release for us there. And if you have if you have heartburn or stomach ulcer or something like that, what do you think your doctor's going to tell you to cut back on? Coffee, anything caffeinated. Also those Red Bulls and stuff along that line. So none of that stuff is going to be good for you because it's going to stimulate acid production and therefore acid release. They also give you medications to block acid production. They're called proton pump inhibitors. So isn't that interesting? They gave a name indicating their actual mechanism. So a proton pump inhibitors is specific for what cell? It's very specific for the parietal cells. So proton pump inhibitors are specifically inhibiting parietal cells production of hydrochloric acid. Also will end up inhibiting production of intrinsic factor. You inhibit a cell, you cause a lot of additional problems when you can't do its normal job. Going down, we're next in the intestinal phase. So this is interesting. The intestinal phase is the opposite of the cephalic and gastric phases, because what is going to happen here is that we're now allowing food, the acidic chyme, to get into the small intestine. Well, the stomach is covered all over with goblet cells, and if we want to have an absorption of our materials, the nutrient materials of our foodstuffs, goblet cells aren't going to do that for us, correct? We need to actually have columnar cells with those little microvilli on their surface. And so we're going to have to realize that when we dump acidic chyme into the small intestine, we actually do it in very small amounts. It only gets a 3 cc squirt of acidic chyme with each little peristaltic or contraction wave of the stomach that's actually going to allow that into the first part of the small intestine or duodenum. So that acidic chyme is going to enter into the duodenum, and we also have chemoreceptors and pressure receptors in that area also. And guess what these are going to do, these chemoreceptors and pressure receptors, when we start putting acidic chyme into an area that's not designed for acid? It's going to tell gastrin to do what? Decrease gastrin production. So that's why this is the opposite, or opposes the cephalic phase and the gastric phase. We want gastrin release to be inhibited. So let's take a look here and add some notes. We need to protect this area from acids. Also, we're throwing in our activated protein digesting enzyme. And so this release is inhibited by distension of the duodenum, because that's where we are into now, which is the first part of the small intestine. And the pH, the low pH, that is now entering in here. So again, we have mechanoreceptors and chemoreceptors in the small intestine, just like we had them in the stomach, but they're going to cause the opposite effect. We want gastrin release inhibited, so we want to decrease gastric juice production. One of the ways that we actually inhibit gastrin release is because of our mechanoreceptors and chemoreceptors, we will have the release of CCK. Cholecystokinin also inhibits gastrin release. So that's the small intestine will also produce that and cause that release. The other thing that will also cause CCK release is the end of the meal. So I want to, most of our foods are a mixture of what? Protein, carbohydrates, and fats, correct? So we're taking that and we're mixing it all up with acidic chyme and creating that lovely material that's going to now go into the small intestine. And as the stomach empties, what is the last of those 3 nutrients that will typically empty out of the stomach? And you do this all the time in your kitchen when you make vinaigrettes. So as the stomach is emptying, squirt, 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 what's the last thing that will want to go out? Fats. That's right, fats. And so fats also, we also detect fats when they enter into the small intestine. Because that means actually the completion of the emptying of the stomach. So these two will also be strong inhibitors of gastrin release. Gastric motility, stomach usually empties 4 hours after a meal. Fats will delay the emptying. So it'll be 4 to 6, depending upon the individual after that. And you have to realize we're going to use our circular, longitudinal, and oblique muscles to actually churn the food. And it will make it into that acidic chyme. And then after we get a good amount of mechanical and chemical degradation or of our foodstuffs, we will then have, after that, we then get peristaltic waves that secrete 3 cc's of the chyme into the small intestine. Or the duodenum. Small intestine, most of our digestion and absorption occurs here. So it's interesting to realize that the digestive processes that occurred in the oral cavity is small, but a larger amount of digestion is occurring in the stomach with acid and pepsin, but it's still nowhere near as much as what's actually going to occur in our small intestine. So we'll get digestion and absorption here. The large intestine is not actually designed for absorption of much other than fluid and some electrolytes and vitamins, and we'll discuss that in a little while. The anatomy of the small intestine, anatomically, it's 20 feet long in the cadaver and only about half as long, or 10 feet long, in a living person. And why do you suppose that is? Yes, muscle tone. In a living person, we've got good muscle tone. You're nice and alive, and you got autonomic nerve myenteric plexus innervating your innards, and so we have nice smooth muscle contraction, keeping that length actually limited. Then once you pass away, we no longer get that innervation, and it just relaxes, and we get about 20 feet of that. The duodenum is the first part of the small intestine, extends off of the stomach, and is only about 10 inches long. And that's actually where it got its name, duodenum, for 10 inches. The jejunum is the central portion, and it is approximately 8 feet long. And then the ileum is the last, and that's about 12 feet long. It ends or terminates at the ileocecal sphincter. So that tees right on into the cecum, and so it's always easiest to figure out where the ileum is by finding where it goes into the cecum, say this is ileum, and then find the duodenum, and the next part is jejunum. And then there's all this lovely mesentery in your cats, correct? Nice, pretty mesentery in the cats, not so pretty in the human being. Lots of fat there. One of the things that's important about the small intestine, in addition to digestion, this is where absorption occurs. So we need to increase surface area in order for absorption to occur. And so the modifications of the tunics and the mucosa will be for that very purpose. Plicae circularis. A plica is a fold. It's Latin for fold. So circular folds are going to be found inside the lumen. You'll actually be able to see them. It's kind of like the rugae that you see in an empty stomach of a cat. Now we have these plicae circularis, and so that's what it's referring to, that kind of ribbed look in there. Those are the folds. So that's done to increase surface area for us and are visible to the naked eye. You can see them quite easily, especially if you put a scope on in somebody. Villi, however, that was what I was pointing to in that histology view. That finger-like projection, that's a villus. So villi are not something that we actually can see very well. It is going to, in order to see it best, it will require a microscope. But this is a actual mucosal fold that has inside of it CT that possesses blood capillaries and lacteals. Lacteals are the name for lymphatic capillaries associated with the digestive tract. Because of the villi, it does actually, when you look at the inside of the small intestine grossly, without the microscope, it appears velvety. So villi are actual significant structures. However, the microvilli, remember, those are the projection on the columnar cells on their apical surface. That project actually into the lumen. And they have on their, associated with their surface on the microvilli, we actually have surface enzymes for further digestion. And so we talked about in the past, you can have proteins associated with the cell membrane, and that's actually what we're referring to, those microvilli with apical, with enzymes for further digestion. Again, this is just meant to show us the plicae circularis. Increasing our surface area that way. This is meant to show you a villus, where we actually have an actual projection of the epithelium and associated CT, where those blood vessels and capillaries and lacteals are located. And again, here, this shows you individual villi over here, all designed to increase surface area. And then we have the microvilli, which is at the cellular level. And all of these are designed to actually increase our surface area. Yes. Any question about the small intestine? Moving on to the liver. The liver is indeed your largest internal organ. It is still considered an accessory structure, and it is responsible for producing bile. That is the thing that it contributes to our digestive processes because bile is a fat emulsifier. You guys remember what emulsification is? That's what mayo is, right? Mayonnaise is an emulsification of gobs of oil, right? Liquid oil and a little bit of an emulsifying agent, sometimes lecithin and sometimes egg. You can make your own mayo at home. You just take a whole bunch of oil and an egg, or actually you take an egg, and you whisk the egg, and then you start drizzling in the oil, and you keep drizzling it, and eventually you will get a really nice mayo out of the stuff, and that's emulsification. So there's an agent that what we're doing when we're whisking it is we're breaking up into little pieces, and the emulsifying agent coats it. And so we have all these microscopic, teeny tiny little pieces, individual pieces that are suspended now in this environment, and the emulsifying agent does that. And that's what bile does for us. So bile is very important for fats in our diet, fats in our food. Anatomically, the liver in us has 4 lobes that are separated by fissures. How about the liver lobes of the cat? Or is it just 4 or more? Definitely a lot more in the cat. So we have 4 lobes. The falciform ligament, though, is present in us as well as in the cat's liver. So realize it's suspending the liver from the diaphragm and the ventral aspect of the abdomen. In the inferior margin of your falciform ligament, there is actually a structure there, even in your cat, that's called the round ligament. Some of the literature calls it ligamentum teres. It is a remnant of our umbilical vein that's actually running up there through the liver. So it's part of our fetal circulation that no longer is needed because we no longer have a placenta and umbilical cord. So that's what the round ligament is. When we take a look at the liver histologically, we find that it's actually divided up into lobules. So a lobule is a structural thing as well as a functional thing for the liver. We can liken it to a microscopic cylinder of hepatocytes. That are radiating out around, away from the central vein, like spokes in a wheel. The central vein is easiest structure to identify histologically when you take a look at a liver slide. We don't have any here, but it is the largest vascular structure in the liver tissue itself, and it drains, it's in the center of the lobule and will drain into hepatic veins, which will then drain into the IVC posterior to the liver. Around the central vein are 6 structures called triads. Triads are 3 things, and what makes up these 3 items in this triad is a hepatic arteriole, a hepatic portal venule, and a piece of the bile duct. So the hepatic arteriole will contain fresh blood. The hepatic portal venule will contain used blood that is nutrient-rich as well as toxin-rich, because this is blood coming from the digestive tract that is responsible for absorption. And then the bile duct for actual bile transport, which is going to go in the opposite direction. So we'll put nutrient-rich, toxin-rich, O2 pour for the blood in the hepatic portal venule. We'll stop there because it is time for you guys to go. We'll talk more about this structure and how things flow in the liver and how things are processed because of that.